a bit like a, yeah. Nah, who am I kidding? It was more like a, whoa. A little bit of oh, 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 yeah, 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 yeah. I'll tell you, there was a whole lot of that. Oh, 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 yeah, 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 yeah. You know what the best bit is though? 6 months on and we still haven't said a word. Well, I tell you right now, singing, I tell you right now. Well, I tell you right now, we've had some mighty fine conversations. Before the first mobile, their plans were set in stone without relying on a telephone. You had to commit when you made a plan, that was it. 'Cause there was no way for your friends to reach you if you didn't show. So give me your word, give me a guarantee that what you say will be an eventuality. Yeah, give me your word. Won't you give me a guarantee that what you say will be an eventuality? Times have changed. We don't put a pencil to the page, and words upon a screen are easier to erase. We didn't think about this. We didn't think about what we missed. Now it's easier to excuse yourself than to commit. So give me a word, you give me a guarantee that what you say will be an eventuality. Oh, yeah, give me a word. Won't you give me a guarantee that what you say will be an eventuality? She's so fine, just like a fine red wine. You know it's a crime to see her home alone. Well, let me tell you all the story. I got something to say about this little slice of heaven I met the other day. See, I've been thinking all about her almost all the time. To be completely, totally honest with you, I don't mind. She's the girl next door. When she knock on my door, she's my Billie Jean lover when we get on the phone.
Good evening, everyone, and welcome to Identity is Everything. A big welcome to everyone joining us on YouTube and Facebook for Pint AU Online. It's Monday, the 11th of May, 2020, and in case you missed it, we are in the middle of a pandemic. So you might be used to attending Pint of Science Australia down at the pub, but tonight we're delighted to be bringing the pub to you. Well, we're bringing science into your homes, just without the alcohol. Maybe a pint of tea that we're celebrating with tonight, but you can have a pint of whatever your choice is this evening. So thank you everyone for joining us. Before we go any further, I'd like to do an acknowledgement of country. So when we usually hold Pint of Science events at pubs around Australia, we like to acknowledge the traditional owners of the land on which we meet. Now that we're holding this event online with people tuning in from everywhere around the country, we'd like to acknowledge all traditional custodians of this land, the first innovators and scientists. We pay our respects to Aboriginal and Torres Strait Islander elders, past, present and emerging, and we recognise their continuing relationship with the land, waters and culture. We'd also like to acknowledge any Aboriginal and Torres Strait Islanders who are attending this evening as well. I'm Dr Brad McKay. I'll be your MC for this evening. I'm a GP and I work in Sydney. You might know me from Embarrassing Bodies Down Under. I did that a few years ago. It was Logie nominated after my social media hijacking, maybe we'll call it that. I've also been on a Catalyst episode recently talking about teeth at a dental clinic as well. And you may also know me from my dog's Instagram account at Mr. Humphrey. You're very welcome to follow my toy cavoodle. He's lovely. So thank you for joining us all tonight. We have some amazing speakers for you. And like me, you'll probably all have questions about immunology because that's really the theme of tonight. It's the opportunity to ask those questions to the professionals working in the field. And you can type your questions in the comment box on YouTube and Facebook, just underneath the video stream, or also on Twitter if you're adventurous with the hashtag PintAUpanels. So the aims of Pint AU Online are just the same as going to the pub. So we're going to be having 3 talks. We'll have 5 minutes for each person. And then afterwards, in our relaxed environment, holding our pint of tea or alcohol or beverage of choice, we will then be asking questions. So put them all through. We've got a team of people going through. And we also have Amy Chan, Research Officer at the University of Queensland. And we've got everyone lined up here. And hopefully our screen is working. I might just ask our guiding light. I have an ethereal voice, Tom, who's with us at the moment. So is everything working according to plan? Yes. Oh, I just don't have a screen, so that's fine. We'll continue on and persevere. So first of all, I'd like to bring Catriona to the front. She is a singing scientist. She sings in the lab and dreams up immunology experiments in the shower. So she enjoys sharing her work. She's been a finalist in the Fame Lab Australia, that was last year, and she's a science communicator. So she's going to be speaking about self-defense or self-attack. So I will hand everything over to Catriona tonight.
Alrighty, thanks, Brad. So I study T cells, and these are specialized immune cells that are one of the main drivers of defense against infection. And they act like security guards, patrolling the body to determine whether the molecules in our cells are self or non-self. So whether they come from our bodies or from other invading bacteria or viruses. And the types of molecules that T cells recognize usually come from proteins, but I study T cells that recognize molecules that come from broken down fats and oils. These aren't as widely studied. We don't know as much about them, but so I refer to them as hipster T cells because they're not conventional T cells and a little bit different from the mainstream ones. When T cells detect molecules that are non-self, so they're coming from bacteria and viruses, the T cells become activated and they'll either go in for the kill, so they'll kill infected cells to limit the spread of infection. It's a little bit like quarantine. Or T cells can also sound alarms to the rest of the immune system and coordinate the entire immune response. And these hipster T cells play a very important role in infection because some bacteria and viruses are absolutely covered in fats. For example, the bacteria that causes tuberculosis or TB is covered in unique fat molecules. So I'm looking at how our immune system recognizes these fat molecules, what they recognize, and how we might be able to add them to a vaccine to boost our immune response. Currently, the TB vaccine isn't very effective, but no one's really tried to target these hipster T cells before. So I'm thinking that if we can manipulate them, we might be able to get better protection. But sometimes T cells don't get that difference between self and non-self quite right, and they'll start to attack our own molecules. And when T cells make mistakes and they think that something's a threat that's really not, it can cause allergy. So for example, these hipster T cells can contribute to allergies to bee and wasp venom. So the bee and wasp venom changes our own skin oil. So you might notice that you have a lot of oil in your skin. So if these are altered, T cells start to attack them because they think that your skin's infected. So I myself suffered a really bad allergy to sunscreen a couple of years ago. And being an immunologist, instead of staying away from the sunscreen like a normal person would, I did some detective work and wanted to know what was going on. And I identified the ingredient that I was allergic to, and coincidentally, it was an oil that is targeted by the very T cells I work on. So now I want to know why some sunscreens cause allergic reactions and how we can stop them. I've found a few ingredients in skincare products that activate T cells and cause allergy. And now I'm researching how this happens and whether our T cells are directly targeting the oils in these skincare products or whether these products are somehow changing our own oils and causing an immune response, just like with the bee and wasp allergy. So there you have it. The immune system is a double-edged sword. You want it to fight against infectious diseases, but not be so strong that it starts attacking you. Then no one should have to think twice about putting on sunscreen, especially in Australia. So to sum this all up, I have a little bit of a song. Patrolling your body are all these T cells. They check the cells of your body to see if they're well. Looking for molecules that don't come from yourself. Things from bacteria and viruses, whatever's non-self. T cells attack when they see something off. They call on immune cells and the immune response kicks off. Some of our T cells attack bacteria that causes TB by recognizing certain fats that just might be the key. Some recognize oils in sunscreen causing allergy. They mistake the oil for a threat and call the cavalry. T cells attack when they see something off. They call on immune cells and the immune response kicks off.
Thank you very much, Katrina. So I will ask you a question at this stage. Have you found a sunscreen that works for you that doesn't give you an allergy?
Yes, well, that was sort of the thing. I lived 20-something years of my life using sunscreen and been fine, and then suddenly I experienced this allergic reaction. And basically now that I know the ingredient that causes it, I can look at sunscreens and say, well, I'll use this, and stay very, very clear of these.
Check the bottle. You don't just need to use a parasol.
Yeah.
Well, thank you very much. And a reminder for everybody, if you're on YouTube or Facebook, please put your questions below and we'll try to get to all of them. We already have a few questions coming through, so please send them through, and we will go on to our next speaker. So our next speaker is Ray Leng. She was appointed the Western Australian Premier's Early to Mid-Career Fellow. Her main research focus is on fingerprinting biological fluids, sounds sticky, to better understand the influence as I get the right screen up, better understand the influence of food on health and diseases. So her talk today, 5-minute talk, is urinary fingerprinting. What is the point? I will hand everything over to Ray Leng.
Hi, thank you, Brad, and for the introduction. So as Brad mentioned, I'm going to talk to you about pee. So have you ever considered what happens each time you flush your toilet, each time you pee? Well, in our urine, it contains a lot of information. So going back to even 15th century, where physicians, they use the urine sample to help them to diagnose in different type of disease. So they even taste the urine, and if they find that the urine is sweet, then this individual will need to change the diet a little bit. Otherwise, they will become really unwell and die. Although at the time, they don't know why and what causes that. But now we know the urine is sweet because it contains sugar, and these are the individuals who are diabetics. So other than tasting the urine, they also look at the colors of the urine. So even modern medications such as drugs that used to treat TB, rifampicin, it can change the color of our urine. And sometimes the food that we eat will also change the urine. So as you can see, our urine tells us a lot of information about the individual, about the disease state, about the food that we eat. So this comes to my research area. So I use a lot of different types of biofood, and urine is one of them. So I'm going to tell you about studies that I have been involved in, and that's called the INTMAP study. It involves around about 5,000 participants, and these are the individuals that recruited from UK, US, China, and Japan. We get a lot of information from these individual participants. We ask them what they eat, what sort of medications that they take, and what sort of exercise they do, how much alcohol they drink, and a lot of information about how much they earn, all sorts. In addition to that, we also ask them to give us urine sample. So I'm glad to say that I don't have to do tasting of urine or smelling them or anything like that, but instead I have different type of instruments that I can use to analyze the urine sample. So one of the instruments that I use to analyze the INTMAP sample is called the nuclear magnetic resonance. And Tom probably will help me to show a picture of what an NMR looks like, a nuclear magnetic resonance. And hopefully when he's ready, he will show that off.
Yep,
and
that's
then,
live. Yep.
Yeah.
Keep going. Yep.
Okay. So what it is that we analyze the sample and we see loads of fingerprinting information. You will see that on the slide that the red spectrum is loads of pigs, which I zoom in so that you can see them. So all these different pigs will show you, will tell us about the different chemical compounds in our urine. And you probably will see that that's another urine fingerprints, which I color-coded that's blue, and it looks very different to the one that is red, just visually by eye. Why? Well, the one that is blue is individuals that are diabetics, and they are full of sugar. All you can see is the sugar molecules in the urine, but not too much anything else. So going back to the INTMAP study, what I did was that we generate all this fingerprinting information from every individual, and we looked at the patterns. But because it contains so many different urine fingerprints, instead of looking at it by eye, we built mathematical model so that we group them fingerprints that look very similar together compared to those that are different. So by just looking at these urine fingerprints, we can tell that people that live in the Western country, for example, UK and US, they're very different from those who live in China or Japan. And not only that, we can also tell the difference between where they're coming from. So you might be wondering, what causes that? Well, one part of the major reason was that diet that we eat is very different. We know what we eat, and it can be sometimes excreted in our urine. So I'll give you some example. Individuals from Japan, because they eat a lot of sashimi, and we can see fingerprinting information in the urine that consists a molecule called TMAO or trimethylamine N oxide, and is related to the fish that we eat. Not only just the type of food that we eat, sometimes the type of alcohol that you drink as well. Again, the Japanese population show a slightly different metabolites in the urine that they in the alcohol that they've been drinking, because they drink sake, which is slightly different to the alcohol that people drinking in the US or UK, for example. So it tells us information, what sort of food that you've eaten or the drinks that you have drunk. Other than the food itself, we also have information about the individual health status. I've mentioned to you earlier on that if somebody who's diabetic, you will see loads of sugar molecule in there. But other than diabetics, we also see other signatures that I didn't expect initially. For example, we know that certain Oriental populations tends to have genetic metabolism differences. So we saw certain molecules were more prominent in the Chinese population, such as the beta amino acid urea condition. Other than the disease itself, we also saw that some of these fingerprinting informations are linked to the high blood pressure. Example of those is like hippuric acid is another molecule that linked to the food that we eat, and eventually is being metabolized by our gut bacteria before it's being excreted out in the urine. So as you can see, overall, by just fingerprinting our urine, it tells us information about your health and the food that you eat, if you've been doing exercise, for example. So my overall research over the next few years as part of my research fellow is that to try to understand what it is that we eat and how that's going to affect our metabolism and how it's going to reflect in our urine fingerprinting that I show you. And hopefully then we can use that to modulate and to make us more healthy again, because we can tell if you're eating a certain type of diet, it might be better for you. So that's pretty much a very quick summary of what my research area is.
Okay. So, well, thank you, Relling. I might ask you a question at this stage as well. So with your research, is it aiming at having sort of a screening test for people that we could, it's like those futuristic machines where you just pee into a cup and it tells you what your future is and every condition that you might possibly have in the future? Or is it more to sort of guide our diet to improve things as we go along?
Yeah, because the technique is actually really versatile. It's not just detecting the type of food signatures that you can see. It's actually can tell us different type of disease state. We'll also have different type of fingerprint information. So at the moment, we're still in the phase of research. We're trying to understand more about our metabolism, because the urine actually you might be surprised to find that it's very complex. It has loads of different molecules in there. And what we know so far is still relatively limited. So I'm hoping to build a bigger picture understand more about the food we eat, and hopefully further down the line in the future, it is something like what you say. If you pee in the urine and the urine will tell us, hey, you have too much sugar in your urine, you better go and see your doctor and check if you're diabetes. Or there's another biomarkers or molecules that we know that link to a disease that can give you early warning sign, so that it can help us to get a better health, really.
Yeah, be great for people who hate blood tests,
Yeah.
and much more accurate than me with a little dipstick into the urine at my clinic. So that sounds like very, very interesting. So for those people who've tuned in, or if I blanked out earlier on and left your screens briefly, just a reminder that you can put questions below the screen, and also if you're game, you can get onto Twitter and use the hashtag #PintAU panels as well, and so we're checking all of these platforms for questions, and so we'll get to them at the end. So we will go on to our final speaker of tonight, which is Amy Chan. So she's recently submitted her PhD thesis, yay. So her research focuses on how our bodies initiate and control inflammation when we get bacterial infections. Amy is passionate for gender equality and cultural diversity in STEM, so thank you very much. Yeah, her talk today will be on fighting bacteria with cellular machines. So I'll hand everything over to Amy now.
Great, thank you, Brad. So I work at the Institute for Molecular Bioscience under the supervision of Professor Schroeder. Our group is called the Inflammasome Lab. So we specialize in studying inflammasomes, which are these protein.
So we've been having some technical difficulties, just like it happens at the pub. The microphone's gone off, but yeah, we're back on board now. I'd like to introduce Amy Chan to talk about fighting bacteria with cellular machines.
Right, thank you, Brad. So I'm a research officer at the University of Queensland. So I just recently submitted my PhD, and hopefully I can get conferred sometime this year. So I currently work at the Institute for Molecular Bioscience with Professor Kate Schroeder, and our group is called the Inflammasome Lab. So we specialize in studying inflammasomes, which are these protein complexes within our cells that drive inflammation. So I'm certain that everyone in this audience has experienced inflammation at some point in your life. I'm sure you're familiar with the sensation of pain, redness, swelling, and heat that results from catching the cold or toe or getting a ear infection. But despite this being quite uncomfortable, inflammation is actually a very protective response. One of the major aims of inflammation is to bring in immune cells like white blood cells to the area of concern. And the other aim of inflammation is to inactivate or to destroy invaders, and also to begin repair in the infected area. So my group studies inflammasomes, which are these protein complexes inside cells. Inflammasomes, like their name suggests, drive inflammation. In my PhD, I work with a very special type of inflammasome, and it's called caspase-4, just this little red dot over here. And this instigates bacterial inflammation. So caspase-4 lives inside our cells. So imagine we're in a cell right now. We have some sort of infection, and now we have bacteria that's in our cells, right? So in this case, caspase-4 can actually sense and detect bacteria and bind to it directly. And this interaction causes caspase-4 to activate. And active caspase-4 can do two things. So the first thing is it can send out these chemicals like PP. So appreciate that the screen here is not mirrored. So it can send out these chemicals to activate other pathways and drive inflammation. The second thing that caspase-4 can actually do is it can cause the cell to explode. So this explosion kills the cell, but it also kills the bacteria that's replicating within the cell. So this whole pathway drives bacterial inflammation and ultimately resulting in the clearance of invading bacteria and induction of tissue repair. Thus, caspase-4 protects us during bacterial infections. However, in some cases, if bacteria manages to get into the bloodstream, infection can quickly disseminate through the entire body. And in this case, infection moves from being a localized infection to a systemic infection called sepsis. So in these cases, inflammation can become overwhelming, and the inflammasome is overactive in this case. The overwhelming inflammatory response can start attacking the body's own cells, and this can cause organ failure, and this condition is called septic shock. Septic shock is a major killer of bacterial infections and has a mortality rate of approaching 40%. So there's currently no cure for septic shock, and there's only supportive care at the hospital at this stage. So inflammation, like Kat said, is a bit of a double-edged sword. So on one hand, not enough inflammation can lead to disease susceptibility and infection. On the other hand, too much inflammation can lead to septic shock in this case. So gaining mechanistic insight of inflammasomes and caspase-4 works is important for us to know ways to control inflammation. So currently, I'm trying to determine how caspase-4 actually turns on and how it becomes active when it senses bacteria, and to characterize its activity after it's turned on. Very importantly, I'm also trying to figure out how caspase-4 switches off. So if we can discover how caspase-4 is switched on and off, maybe we can control its activity to ramp up inflammation in the initial stages of bacterial infection, or to reduce its activity if it gets out of control. So hopefully this will lead to development of new drugs and therapies in the future. Thank you, Brad.
Cool. Well, thank you very much, Amy. That is fantastic. I think everyone has been talking about oh, sorry, I got my voice in my head. So I'm not psychotic. It's just an echo. Yeah, like a lot of people are talking about inflammation with coronavirus at the moment, and a lot of people are talking about septicemia and septic shock. It's sort of like known a little bit more these days compared to a decade or so ago. A question for you now is sort of like, are you finding that there are amazing, like promising treatments to sort of cut off that inflammation that we could possibly use to stop that infection or stop that septic shock at the moment?
Actually, that's a very interesting question, and it's a very timely question at the moment. There is actually a company in Ireland called Inflazome that is using inhibitory drugs to target a different type of inflammasome. So not specifically one I'm working with, but a different type of inflammasome that is actually central to a lot of different diseases in humans, including cancer, neurodegenerative diseases like Alzheimer's and Parkinson's. And this drug is currently undergoing human trials. So hopefully we will see more of it in the future. Definitely a very hot topic at the moment, and many labs around the world are looking at targeting inflammasome activity with drugs.
Yeah, cool. Thank you very much. So so we will be taking your questions. This is like the best part of the night, where we sort of get to hand over to you. So please send in your questions on YouTube or Facebook, or else you can also go on to Twitter at #PintAU panels. So we have somebody monitoring that at the moment, and they're very lonely. So please send them messages, so they can respond and send everything through. So so we'll sort of like we'll go backwards and forwards a little bit. I think the first question that's come up here, where am I, is really directed at Catriona. So this is from Leslie Erin on Facebook. So she said that I've had the exact same experience with sunscreen, so that allergic response. I haven't been able to narrow down the ingredient though. What ingredients have you found are interacting with T cells? So of course this is not medical advice, but yeah, like I'd certainly be interested in your opinion or your experience with this, Catriona.
Yeah, as Brad said, I can't give medical advice. I'm definitely not qualified to do that, but and there are different causes for allergy as well. What I'm studying is one cause of many, but in terms of these hipster T cells that I'm working on. Another group over in Harvard, who are our collaborators, they've actually already published their findings. They've found that compounds like that have benzyl rings, so that's a little bit of structural chemistry for you, but if it has benzyl in the name, they can be presented to these T cells and be targeted. So that's one of the ones that I try and avoid. If I see benzyl in a thing, I'll avoid it personally, but like I said, you could be different. So
yeah, so it's trying to identify that ingredient and trying to avoid it if you can. Of course, like seeing a dermatologist, me being a GP, yes, go and see a dermatologist, and sometimes we will be able to do allergy testing as well. So sorry, I'll just pop my my medical hand up there. So the next question here is from Audrey on YouTube. She says, "Hey, Raylene, hi. So how unique is an individual's urinary fingerprint? Will it be possible to ID people specifically, similar to DNA or hand fingerprints?"
Well, it's not as specific as such, because it gives us if you have a genetic defect that makes your metabolism that's slightly different, then it will show up. But again, we won't be able to say that this particular urine is from you, Brad, for example, or so that is a little bit difficult to do that. But as a whole, we can generate a lot of information about the individual rather than like genetic, where we can say this is you for sure, really.
Yeah. So could you eat asparagus to copy somebody else's urine, for example, and pretend to be somebody else?
I think that would be difficult, because again, all our gut bacteria could be very different, and that will be influenced by what you eat. If you are a vegetarian, your gut bacteria would be very, very different to somebody who is eating meat all the time. And so those gut bacteria will also show certain signatures in your urine. So even though you're suddenly trying to mimic somebody's diet, your gut bacteria signature might still be changing until, although having said that, it can change quite quickly the minute that you start changing your diet. So I think at the moment it's difficult to be able to pinpoint saying that this urine is you, for example, unlike in genetic analysis, really.
So I can make it smell like somebody else, but wouldn't be showing up the same on the machine.
No. Yeah.
So the next question here, so this is for Amy. So and from Alison on Facebook, she says, "Hi Amy, I'm curious about how many diseases result in inflammation. So is it every disease?"
I'm sorry, Brad, you cut out a little bit. Was it how many diseases result in inflammation?
So, so the question's from Alison. Sorry, there is a delay on the line. So Alison is curious about how many diseases result in inflammation. So, is it every disease? So, it's a very general question. So, I'll hand it over to you.
Right. Sure. So right now with inflammation is a bit of a chicken and the egg question, because we are now starting to understand that inflammation is actually the driver of diseases rather than the cause, rather than the symptom of disease. So many different types of diseases can have an inflammatory response or inflammatory symptoms to it. But as we're now starting to understand, the chemicals sent out from inflammation is actually also causing physiological changes within your body. And it can lead to cancers and arthritis and different neurodegenerative diseases as well. So right now, it is at the moment a very highly researched topic is whether inflammation comes before the disease or whether it's just a symptom of disease. That being said, I think it's very important for us to still remember that inflammation is a very important protective response of the body, and it helps us clear infection and to repair damage to cells. So it's not necessarily a bad thing to drive these inflammatory symptoms. It's just when inflammation goes out of control or if it's overwhelming that it causes diseases.
Okay. So our next question is, where are we? For Katriana from YouTube. So is the mechanism involved in sunscreen allergy the same mechanism that causes hay fever or bee sting allergies?
Good question. Short answer is no. So there are different types of immune cells. So I look at T cells, and then there are also B cells. We just like letters. And lots of other types of immune cells as well. So hay fever is a much more rapid response, and it's caused by a different kettle of fish, whereas T cell responses tend to be slower. So if you are getting a skin allergy that is caused by T cells, usually the first time you use something, you'll be fine, and you're exposing yourself to, whatever it is that you're allergic to, and then it's the second time and the subsequent times, that's when you're starting to get the allergy. And that's what made me realize I had a T cell response personally. And so hay fever and skin allergies are quite different. And in terms of bee allergies, that would be different again. But as I mentioned, part of bee allergies can be caused by the immune cells that I work on, and that would be the same mechanism. The immune system is complicated.
So I'll throw you a curly question for myself as well. There's a weird condition called cholinergic urticaria where people just get an allergy, like they get a red rash, the same as having a bee sting allergy, but they get it from like a change in weather, like from hot or cold, or they might get it from like tasting chili or something else. It's triggered. Are you very conscious about what causes that sort of like strange immune response to happen from something that would be usually quite benign?
Yeah, not exactly, but I do notice that for some people, sweat in particular can trigger it. So I guess if you're in hotter weather or if you are eating chili, you probably are going to be sweating. So I can't explain that, but I am aware that sweat does trigger that as well, because it's sort of changed. Potentially, if you're looking at it from my researcher's perspective, you are changing the oil composition of your skin.
So it could be something that's switching over that oil composition into another sort of substrate that then could be causing an allergic style response.
Potentially, but I couldn't comment on that condition particular.
Thank you. I'm just interested myself. So thank you very much. So the next question for Reiling. Where are we? This question is from Kevin Law from YouTube. Can you please explain more about how exercise metabolically changes the readings you get from urine samples?
Depends. Sometimes there's currently studies going on. That depends on whether you are doing some of those low intensity or versus high intensity exercise. It can affect that. So one of the more classic one is a marker called creatinine that will be easily identified in the creatine that can easily be identified using NMR, because once you've done exercise, is a muscle breakdown, then you'll be able to see that particular peak in the urine sample. And there are other instruments that can be used to measure urine sample as well. Again, there are more different type of fingerprinting information that generate from that. But again, we're still trying to figure it out more. So how does that overall can affect our signatures really in the urine sample?
Yeah. So okay. The next question here, so we'll try and race through as many questions as we can. Another one for Reiling. So how long does it take for these tests to yield results? Do you think that they will overtake current testing for diabetes or other diseases?
Currently, we still use, as Brad mentioned, we would just use a dipstick test. It's a very cheap and efficient way. And the methods that we're using is actually quite quick. So once we have to prepare the sample, just running one of the experiment, it just takes less than 5 minutes. So depends, and there are sometimes we analyze different type of biofluids, such as the blood sample. Again, we can measure loads of different type of lipoprotein within 10 minutes, and that's have more types of subtypes of lipoprotein compared to the typical classic clinical measurement that is available in the GP practice, really. But having said that, these methods, a lot of them are still in research development. They're not quite used in a GP practice scenario yet, but the method that we use are quite high, is very high throughput, and it can use to measure loads of sample in a high quantity, really. So maybe in the future, it is something that we can move towards, but obviously, because as you can see earlier on from one of my general urine sample, it contains loads of different peaks, and it's just not very easy to get a GP to look at those fingerprints and know what that means, really. So what we will probably want to do is that in the future, generate those mathematical models where you can actually tell very quickly if this is the pattern, this means that this individual is having a diabetes. And in fact, some of the other instruments have been used in clinical trial setting. For example, one of the mass spectrometry instrument where they actually use it in a theater environment where the surgeon cutting out the patient's tumor cell, and they are actually analyzing it at the same time. As they analyze the sample, they put it through into the mathematical model, and they can tell that actually it contains tumor cells related type of molecule, which typically these are fat related type of molecules, versus if that's a healthy tissue. And in those scenarios, they actually can tell in a very short couple of minutes or so, very high, very much interactive, and it gives the decision for the clinicians to know whether if they should continue cutting that piece of tissue or actually they reach the healthy tissue cell. So these are some of this research work that's been currently running or being tried out in some of my colleagues or different researchers all around the world using this technique. Some is more advanced than others, but a short answer is that it's not quite get to that way yet, but ultimately maybe developing a kind of like a dipstick equivalent type. If you want to check this particular condition, dip it in there and it will tell you whether it's contained urine very quickly, rather than all this complicated fingerprinting informations, really.
So how much does your machine cost, and can I put it at the end of my clinic desk?
The machine itself is actually quite expensive, the one that you saw, which is the 600 megahertz instrument. So the higher the frequency that is, it will tell you more information. And I think it was around 700, 800K per instrument.
Okay. So I'm not going to put it in my clinic at the moment.
You're not.
No.
What you want to do is that you can send sample through to our research laboratory, and to help to analyze that, to run those samples is relatively cheap, actually.
I will spare you from sending you all of my piss at the moment.
Actually, we love having all this sample, to be honest.
We may put up a link so people can send you lots of urine. So,
yeah, when we do recruit volunteers, maybe we'll let you know.
Yeah, definitely. So our next question is for Amy as well. This is from Fifi on YouTube. And this is a really good question. What is a cytokine storm?
A cytokine storm is when immune cells are sending out too many of these cytokines. So cytokines are these signals, these chemical signals that I talked about a little bit earlier, these little pew-pew signals. And cytokines, well, there are many different types of cytokines. Some of them induce tissue repair, some of them produce pain signals, and some of them induce fever. And all of these cytokines have different activities on different sort of immune cells or different cells in general. A cytokine storm is when you have an overwhelming immune response. So all of your cells are sending out these chemical signals and then basically activating a lot of immune cells, a lot of different types of chemical responses in your body. And this can actually be very damaging, because when all of this activity is happening in your body, it can actually start damaging your organs. It can actually start causing a lot of cell death. And that's why cytokine storm is actually a very large killer for a lot of diseases, including the flu. And yeah, a lot of mortality for flu and viruses is caused by cytokine storm.
Our next question here is for Catriona from Levi Barker. So how and why do our immune cells attack our own body cells in autoimmune conditions? So especially like those in Guillain-Barré syndrome, which are caused by bacterial infections.
There are different reasons that your own body can start to attack itself, but you've just brought up a really good example in that sometimes the molecules that T cells see are very, very similar between bacteria and our own, because everything basically comes down to shape. And if the shape is very, very similar, then sometimes your T cells will get on with fighting the bacteria or the virus. I say virus as well because that's sometimes the case for type 1 diabetes. So they clear the bacteria or the virus, and then you've got all these immune cells that are geared up, ready to fight, and then they turn around and they start to see, so I'll continue with diabetes as an example, they then see your pancreatic beta cells, and they're like, oh, it's the same thing. It's the infection, let's get rid of them. So then they start to kill the cells in your pancreas that create insulin. So that's sometimes why people get type 1 diabetes, and it's a very similar process. It's just the fact that things look too similar.
Mm-hmm. I suppose on that theme, with Guillain-Barré syndrome, for those people who may not know much about it, it's sort of when there could be a viral infection or a bacterial infection. And again, it's what you're saying, Catriona, where your immune system identifies a part of your body as looking similar to that infection. And in this case, it seems to be like the surroundings of nerves, so the sheath that's around the nerves. And then people end up having paralysis in different areas of the body, wherever that immune response is from. I suppose you may not know the answer to it, but why would it just affect some nerves and not others if your immune system is sort of everywhere? And why would it just be transient? Because a lot of people with Guillain-Barré syndrome would only like have paralysis maybe for a week, and then they'll slowly improve over time. I don't know if that's stretching the boundaries of your knowledge.
A little bit, but I can have a go. We have different sort of, we call them microenvironments. So you have different environments within your body depending on the surrounding cells. And a big group of cells that try and prevent autoimmune reactions are regulatory T cells. So you do have T cells that kind of keep everything else in check. So it could also potentially be a balance of how many of those you have around in particular areas. But it might not just be the regulatory T cells. It probably is also influenced by what other cues and environmental sort of senses cells are getting.
It's far too complex. This is why I find immunology very difficult. So, but vast concepts is fine for me. So the next question here, this is for, where are we? I might ask Amy. Amy, can caspase 4 attack non-bacterial microbes like viruses or parasites? This is a question from Kevin Law on YouTube.
Can caspase 4 bacterial microbes?
So can caspase 4 attack non-bacterial microbes? Can it also attack viruses and parasites as well?
So caspase 4 cannot attack viruses and parasites. It's very specifically bacteria that it targets, and it only targets gram-negative bacteria. So if we're talking about different bacterial classifications, bacteria are usually classified into gram-negative and gram-positive. And the reason caspase-4 only targets gram-negative bacteria is because gram-negative bacteria have a little structure within their cell wall called lipopolysaccharide, or LPS. So caspase-4 specifically recognizes lipopolysaccharide, and that's how it can dock to the gram-negative bacteria and induce its activity. Viruses and parasites don't express lipopolysaccharide, and so caspase-4 cannot recognize viruses and parasites. So caspase-4 is actually very specific of what it targets.
Cool, thank you. And we've got time for a couple more questions. So for Ray Lang, I love this question. This is from Jirana on Twitter. Could urine become a new way for crime scene analysts to analyze crime scenes, similar to how CSI has shown us with fingerprints? It's sort of hypothesizing the future.
I guess it's possible in the future if we know more about the metabolism in human and understand more how that, I guess that's kind of related back to one of the questions previously, whether we can tell it definitively it is somebody's gene using, or that is somebody urine using the fingerprinting information. I guess maybe at the moment we're not quite there yet, but maybe in the future when we understand the human metabolism, the human body a little bit more, and who knows?
A very, very good answer. So, two more questions, I think, at the moment. So for Catriona, so are these hipster T cells having an autoimmune response to lipids produced by our cells outside of infection?
Yeah, potentially. So, sorry, the question is attacking our own self-lipids outside of infection?
Yeah, so are the hipster T cells having an autoimmune response to lipids? So not related to infection at all. So our own immune system, our own autoimmune system.
Yeah, so we've seen that these hipster T cells can actually recognize our own skin oils. So I talked about the bee and wasp and changing our skin oils, but even without that, our normal skin oils can be recognized by these T cells. So that's one context, and also looking at lipids, or for people who don't know what lipids are, these fat and oil molecules, looking at them in the nervous system. There are a lot in the nervous system, and we're looking at adipose tissue as well. Some of these actually can be picked up and shown to T cells, and some T cells can indeed respond to them. So we do know that the numbers of these T cells might change during autoimmune conditions, such as those sort of central nervous system ones, but we haven't yet necessarily linked these T cells with those conditions as causative.
Okay, yeah. So, and we'll finish with our final question, which is for all of the panelists here, and this is very common for me as a GP as well. So as immunology experts, what is your best snappy retort when someone claims they have a product that can boost your immune system? This is from Cherry Black on YouTube. So who would like to answer this?
Don't dial up to 11.
I'd say...
So if I'm telling you that echinacea can boost my immune system and prevent me from getting coronavirus, what would be your response to somebody saying that? So Ray Ling, I'll pose that to you.
Well, I guess then I will need to understand more a little bit about that, and what particular molecules that they think it can boost the immune response. And what makes it more interesting is that all of us are very different. Whether if I take it, we'll have the same boosting of immune response versus somebody else, because how we metabolize that will be different, and it's also depending on how much is that in that particular product that you refer to. So it's all currently, I guess the food industry or the food wise, they tend to claim certain benefit, but the evidence behind that at the moment is probably lacking. But it's just saying that, oh, this particular compound contain, I don't know, antioxidants or something like that, that would be good for your immune response. But they never really actually look at how much antioxidant is in there, and is it actually going to evoke a physiological response for that particular individual. So that's my take on that, really.
Yeah, and Catriona, like with that question, sometimes people will say, yes, this is boosting my immune system. What does that statement really mean to you? Like you deal with allergies, so boosting the immune system could just mean that you're getting like a heightened immune response that's horrible. What's your response with that boosting?
Well, you asked for a snappy retort, right? My snappy retort is, if you're not getting constantly infected with things, your immune system is fine. You don't need to boost it. Yeah, you're absolutely right. If you're boosting it, you can start to overreact to things. Like you get allergies, you get autoimmune diseases. So just if you're not getting infected, you're fine. Your immune system's battling everything as it normally should.
Yep, and Amy, I pose this to you as well. So if you're wanting to boost your immune system, would it be better to get measles or a measles vaccine?
Oh my God, sorry, I didn't catch the last part of that.
Sorry, that's the delay on the line. So would you rather, if you're wanting to boost your immune system, would you rather get measles or a measles vaccination?
Oh gosh. Of course, the measles vaccination. So the reason vaccination is so effective is because, well, in a lot of cases, they use an inactivated virus or inactivated bacteria that's not actually going to cause disease within your body. It has your immune cells can recognize and then respond to in a later stage. Which is why vaccinations work, and you should absolutely vaccinate and promote vaccinations. They are safe as opposed to actually catching measles, which can be very life-threatening. So that's my answer.
Avoid cytokine storms.
Absolutely.
Yeah, well, thank you so much, guys. It's been a pleasure to speak with everybody. We will close up here, but yeah, a really big thank you to Catriona Nguyen Robinson, Rayleng Liu, and Amy Chan. Thank you for answering all of our questions this evening. We've covered as many questions as we could possibly get to tonight, and our speakers have also offered, we'll say this, that you've generously offered to answer more questions on social media if possible. Even if you don't get around to it tonight because it's getting a little bit late, if there's any possibility of checking online and answering other people's questions, that'd be really, really good. But thank you so much for your time tonight, guys.
Thank you for joining us.
And for everyone who's watching from home, if you're keen to speak with more scientists on the ground, there's plenty more Pint AU online opportunities coming up. So tomorrow night, on Tuesday, the 12th of May at 7:30 p.m., there is online trivia, and there will even be prizes. So that's going to be pretty good. Maybe it's toilet paper. We'll see how we go. So on Wednesday, the 13th of May, we'll be holding 2 more panels. So the first one is What's the Matter? Featuring speakers on astrophysics, quantum physics, and nuclear fusion with host Kirsten Banks. She's delightful. And there's also Brain Health in Our Modern World, featuring speakers on stroke, dementia, and how the built environment affects our mood. And that's going to be with host Broderick Matthews, who is also delightful. So if you've forgotten both of those, then we'll have a link for you in the live stream description below. You can also check what's going on in This Is My Science. So hashtag on Twitter and apparently on Instagram as well. So hashtag This Is My Science. I've heard that it was 4th trending today, neck and neck with Obama. So that's a pretty good hashtag. This is scientists who are going through threads and talking about the science that they're doing at the moment. So please check that out. Also, if you're into Reddit, then go to Ask Me Anything. So we've had it going today. It's going to be on tomorrow and Wednesday with scientists answering all of your questions. You'll be able to ask me anything. Well, not me, but them. And there's also a podcast, Pod of Science. So this will be short interviews with scientists working around Australia. And for all information, if you just forget everything that I've said, go to pintofscience.com.au/online. So thank you, everyone, for joining us. Thank you to the 250 volunteers and 450 speakers who prepared for the original pub events that unfortunately had to be cancelled because of a weird virus that's created a pandemic around the world. How dare it? Thank you to the small team of about 20 people who have done a huge amount of work over the past month to put these new online events together at the last minute. Thank you so much, including our artist in residence, Keith, and our graphic designer, Nicole, who put together the amazing artwork that you've been benefiting from today. Thank you again to our speakers and the audience, because these PINT events are really from this interaction and having the conversation. This is what's really important. And to everyone watching at home, in quarantine or isolation, thank you so much for joining us this evening. Thank you to the speakers again, and everybody remember to wash your hands.
By the seashore, when it rains and it pours on the video.